In the previous session, we have started the second chapter called the sexual reproduction in the flowering plants, where we studied the structure of a typical flower and the male part of the flower, the structure of the bilobed anther, pollen sac structure. The pollen sac can also be called as the microsporangia, a process called microsporogenesis, where it involves the formation of the microspores, microgametogenesis, that involves the formation of the two male gamete in the pollen grain, structure of the pollen grain, and economic importance of the pollen grain. As we know, the female part of the flower is called gynoecium. The gynoecium can also be named as pistil or carpel. It is made up of stigma, the style, and the ovary. So gynoecium ಅಲ್ಲಿ ಒಟ್ಟು ಮೂರು ಪಾರ್ಟ್ ಇರುತ್ತೆ: ಸ್ಟಿಗ್ಮಾ, ಸ್ಟೈಲ್, ovary. Stigma, it is the tip of the gynoecium, which is sticky in nature and that provides a platform for the pollen grains to land and to germinate. ಎರಡನೇದು ಸ್ಟೈಲ್ ಅಂತ. ಸ್ಟೈಲ್ is slender tube-like structure that extends from the tip of the ovary and it gives the way for the growth of the pollen tube. Ovary. Ovary is the basal part of the gynoecium. Within the ovary, there is a cushion-like structure called placenta. On the placenta, there is the ovule. Okay, ಮತ್ತೊಮ್ಮೆ ಹೇಳ್ತೀನಿ. The female part of the flower is called gynoecium. The gynoecium can also be called as the pistil or carpel. The gynoecium is made up of three regions here. The first region is called the stigma. Stigma is the first part of the gynoecium. It is the tip of the gynoecium. It is sticky in nature and it provides a platform for the pollen grains to land and to germinate. ಎರಡನೇದು ಸ್ಟೈಲ್ ಅಂತ. Style is a slender tube-like structure that extends from the tip of the ovary and it gives a way for the growth of the pollen tube. ಮೂರನೇದು ovary ಅಂತ. Ovary is the basal part of the gynoecium. It has cushion-like structure called placenta. On the placenta, there is development of the ovule. So, the ovule can also be called as the megasporangia. Now, we will study about the ovule. There are around 6 different types of the ovule based on the position of the micropyle. These ovules are: number 1, orthotropous ovule, anatropous ovule, hemiotropous ovule, campylotropous ovule, amphitropous ovule, cercinotropous ovule. Okay, there are different types of the ovule based upon the micropyle, like orthotropous, anatropous, hemiotropous, campylotropous, amphitropous, and cercinotropous ovules. Among these 6 different types of the ovule, anatropous ovule is a typical ovule that can be studied as an overview. Now, we will study the structure of anatropous ovule. Anatropous ovule can also be called as megasporangium. Anatropous ovule is a kind of inverted ovule. It is present in the ovary on a cushion-like structure called placenta. Structure ಬಂದಾಗ, if you look the structure of the anatropous ovule or megasporangium, a structure ಬಂದಾಗ ಏನಿರುತ್ತೆ ಅಂದರೆ, the first part is called the funicle. FUNICL, funicle ಅಂತ ಹೇಳ್ತೀವಿ. So what is a funicle? Funicle is the small stalk of the ovule that attaches to the placenta. ಓಕೆ, ಫಿನಿಕಲ್ ಅಂದರೆ ಏನು? ಫಿನಿಕಲ್ ಒಂದು ಸಣ್ಣ ಕಡ್ಡಿ ಥರ ಇರುತ್ತೆ, ಪ್ಲಾಸೆಂಟಾಗಿ ಅಟ್ಯಾಚ್ ಆಗುತ್ತೆ ಅದು. ಸೊ ಫಿನಿಕಲ್ ಇಸ್ ಅ ಸ್ಮಾಲ್ ಸ್ಟಾಕ್ ಅಟ್ಯಾಚಸ್ ದ ಓವ್ಯೂಲ್ ಟು ಪ್ಲಾಸೆಂಟಾ. ಎರಡನೇದು ನ್ಯೂಸಿಲಸ್ ಅಂತ. ನ್ಯೂಸಿಲಸ್ ಈಸ್ ಮೇಡ್ ಅಪ್ ಆಫ್ ಪ್ಯಾರೆನ್ಕೈಮಾ ಟಿಶ್ಯೂ. ಇಟ್ ಈಸ್ ಏನು ಮಾಡುತ್ತೆ ನ್ಯೂಸಿಲಸ್ ಅಂದರೆ, ದ ನ್ಯೂಸಿಲಸ್ ಈಸ್ ದ ಪ್ಯಾರೆನ್ಕೈಮಾ ಟಿಶ್ಯೂ. ಅದೇನು ಮಾಡುತ್ತೆ ಅಂದರೆ, ದೆ ಸ್ಟೋರ್ ರಿಸರ್ವ್ ಫುಡ್ ಮೆಟೀರಿಯಲ್. ಮೂರನೇದು ಇಂಟಿಗ್ಯುಮೆಂಟ್ ಅಂತ. ಇಂಟಿಗ್ಯುಮೆಂಟ್ ಈಸ್ ದ ಪ್ರೊಟೆಕ್ಟಿವ್ ಕವರಿಂಗ್ ಪ್ರೆಸೆಂಟ್ ಅರೌಂಡ್ ದ ನ್ಯೂಸಿಲಸ್, and the integument may have one or two integuments. So integument is a protective covering present around the nucellus. They may have one integument or two integuments. The next, it is called hilum. Hilum is a point of attachment of the body of the ovule to funicle. Hilum andre eno? It is a point where the ovule attaches to the funicle. Next comes about the chalaza anta. Chalaza is the basal part of the ovule. It is the basal part of the ovule. Here the integument covers the nucellus completely. Chalaza ಭಾಗ ತಂದಾಗ chalaza ಏನಾಗುತ್ತೆ? It is the basal part of the ovule. The integument covers the nucellus completely. The next, micropyle ಅಂತ. Micropyle ಅಂದರೆ, it is a small gap or opening that is left at the opposite part of the chalaza. Chalaza opposite ಕಡೆ ಒಂದು ಸಣ್ಣ ಗ್ಯಾಪ್ ಇರುತ್ತೆ, ಸಣ್ಣ ಓಪನಿಂಗ್ ಇರುತ್ತೆ. This gap or this opening is called the micropyle. Next, raphe ಅಂತ. RAPH, raphe ಅಂತ. Raphe is a small ridge that extends from the funicle ಅಪ್ ಟು ದ ಚಲಾಸಲ್ ಎಂಡ್. ಅದೊಂದು ಸಣ್ಣ ರಿಡ್ಜ್ ಇರುತ್ತೆ, ಅದೆಲ್ಲಿರುತ್ತೆ ಅಂದರೆ, ದೇ ಯೂಶಲಿ ಎಕ್ಸ್ಟೆಂಡ್ ಫ್ರಮ್ ದ ಫಿನಿಕಲ್ ಟು ದ ಚಲಾಸ. ದ ಲಾಸ್ಟ್, ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಅಂತ. ಸೊ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಈಸ್ ಫೌಂಡ್ ಇನ್ ದ ಸೆಂಟರ್ ಆಫ್ ದ ನ್ಯೂಸಿಲಸ್. These embryo sac can also be called as the female gametophyte. The embryo sac are usually found towards the micropyle end of the ovule. Now we will study the structure of the embryo sac. Embryo sac can also be called as female gametophyte. The embryo sac is usually made up of the following types of the cells. Embryo sac ನೋಡಲಿಕ್ಕೆ ಹೆಂಗಿರುತ್ತೆ? ಈ ಥರ ಇರುತ್ತೆ embryo sac ಇದು. So we already find the embryo sac. Embryo sac are found in the nucellus. They are found embedded in the nucellus. There are around 3 categories of cells here. The first category of cell is called antipodal cells. Antipodal cells ನಂಬರ್ ಎಷ್ಟಿರುತ್ತೆ? ಒಟ್ಟು 3 antipodal cells ಇರುತ್ತೆ. Where do you find them? They are found towards the chalaza end. ಎರಡನೇದು, it is called the central cell. Where do you find central cell? They are found in the center of the embryo sac having 2 nucleus. Number 3, it is called egg apparatus. This egg apparatus is made up of 2 synergids and 1 egg cell. They are found towards the micropyle end. The synergids, they have finger-like structure and they are called filiform apparatus. So, ಏನು ಮಾಡುತ್ತೆ filiform apparatus? The filiform apparatus, they guide the entry of the pollen tube into the synergids. I will repeat once again. If you look at the structure of the embryo sac, it is made up of 3 categories of cells. The first category of cells are called antipodal cells. The number is 3. They are found towards the chalazal end. Number 2, ಎರಡನೇದು central cell ಅಂತ. Central cell is a single cell found in the center having 2 nucleus. Number 3, egg apparatus. Egg apparatus is made up of 2 synergids and 1 egg cell. ಅದೆಲ್ಲಿರುತ್ತೆ ಅಂದರೆ, they are found towards the micropyle end. Here the synergids, they are finger-like structure and they are called filiform apparatus. ಇದೇನು ಮಾಡುತ್ತೆ ಇದು filiform apparatus? They help in guiding the entry of the pollen tube into the synergids. Okay, fine. Next, now we'll study about the next concept. It is called megasporogenesis. Megasporogenesis ಅಂದರೆ ಏನು? Megasporogenesis ಅಂದರೆ it is the formation of the megaspore from megaspore mother cell. Megaspore mother cell ಇಂದ megaspore formation ಆಗುತ್ತೆ. This is called megasporogenesis. ಹಾಗಾದರೆ ಒಂದು ಮೆಗಾಸ್ಪೋರ್ ಹೆಂಗೆ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ? The formation of the megaspore from the megaspore mother cell takes place only by meiotic cell division. So, meiosis ಮುಖಾಂತರ ಏನಾಗುತ್ತೆ? There is formation of the megaspore. Now, we will study the steps involved in the megasporogenesis. Steps ಹೆಂಗಾಗುತ್ತೆ ಅಂತ. ಹಿಂಗಾಗುತ್ತೆ ಅಂದರೆ ಈ ಓವ್ಯೂಲ್ ಒಳಗಡೆ, so inside the ovule there are some hypodermal cells. These hypodermal cells initially, okay, they will be very small. As they enlarge, they are called as archisporium ಅಂತ ಹೇಳ್ತೀವಿ ಅಥವಾ archisporial cell ಅಂತ ಹೇಳ್ತೀವಿ. So ovule ಒಳಗಡೆ there will be a number of hypodermal cells. These hypodermal cells they enlarges and they are said to be archisporium or archisporial cell anta heltivi. These archisporial cells they divide into two categories of cells, and outer parietal cells and inner sporogenous cells. So these sporogenous cells they functions as the megaspore mother cell. So out of the two categories of cell, that means parietal cells, sporogenous cell ತಗೊಂಡಾಗ the sporogenous cells they functions as the megaspore mother cell. So what is the function of the megaspore mother cell here? The megaspore mother cell undergoes two meiotic division to produce 4 haploid megaspores. So megaspore mother cell ಏನಾಗುತ್ತೆ? They undergo 2 meiotic division. Divide ಆದಾಗ they are going to produce 4 haploid megaspores. These 4 haploid megaspores they get arranged in a linear fashions and they are said to be linear tetrads. So out of the 4 megaspore, the 3 megaspore they degenerate and the remaining one become active and it develops into a female gametophyte or embryo sac. So therefore the development of the embryo sac from a single megaspore is called monosporic development. Okay, so we will now just summarize what is megasporogenesis here. Megasporogenesis ತಗೊಂಡಾಗ megasporogenesis means it is the formation of the megaspore from the megaspore mother cells. So the megaspores are produced only through meiotic cell division. Steps involvement mechanism ಹೆಂಗೆ ಬರುತ್ತೆ, ಹೆಂಗೆ megaspore formation ಆಗುತ್ತೆ? See, some of the hypodermal cells that remains in the ovules they start to enlarge ಆಗುತ್ತೆ and they are said to be archisporial cells or archisporium cells. These archisporium cells they divide into two categories of cells. They are called the parietal cells and sporogenous cells. These sporogenous cells they behave, they functions as the megaspore cells. So these megaspore cells they undergo 2 meiotic division. ಎರಡು meiotic division ಆದಾಗ they are going to produce 4 haploid megaspores. These 4 haploid megaspores they get arranged in a linear fashion. ಉದ್ದಕ್ಕೆ ಅರೇಂಜ್ ಆಗುತ್ತೆ ಅದು. So out of these 4 megaspore, the 3 megaspore degenerate and remaining 1 megaspore it become active, it become functions, it start to function, and this active megaspore develops into a female gametophyte or embryo sac. Therefore the development of the embryo sac from a single megaspore is called monosporic development. What do we call? We call it as a monosporic development. So now we will study the next concept called megagametogenesis. Megagametogenesis ಅಂದರೆ ಏನು? The process of formation of the embryo sac or the process of formation of the female gametophyte. ತಪ್ಪು ಹೇಳಿದೆ. So now we'll study about the next concept called megagametogenesis. So what is megagametogenesis? Megagametogenesis means it is a process of formation of the egg in the embryo sac is called megagametogenesis. ಹಿಂಗಾಗುತ್ತೆ ಇಲ್ಲಿ ತಪ್ಪಾಯ್ತು. Fine. So now we'll study about the megagametogenesis. So what's the megagametogenesis? It is a process of formation of the egg Fine. So now we'll study about the megagametogenesis. What's the megagametogenesis? It's a process of formation of the embryo sac or the female gametophyte is called the megagametogenesis. So embryo sac, female gametophyte young produce agutte, they produced through a single megaspore. They produced from a single megaspore. So the process of formation of the embryo sac or female gametophyte from a single megaspore is called megagametogenesis. If you look in terms of the steps, ಹೆಂಗೆ ಪ್ರೊಡ್ಯೂಸ್ ಆಗುತ್ತೆ ಅಂದರೆ ಸಿ Now we'll study about the next concept called megagametogenesis. Megagametogenesis andre ಏನು? It is a process of formation of the embryo sac from a single megaspore is called megagametogenesis. So megagametogenesis ಅಂದರೆ ಒಂದು ಸಿಂಗಲ್ ಮೆಗಾಸ್ಪೋರ್ ಏನಾಗುತ್ತೆ ಎಂಬ್ರಿಯೋ ಸಾಕ್ ಡೆವಲಪ್ ಆಗುತ್ತೆ. ಇದಕ್ಕೆ ನಾವೇನಂತ ಕರಿತೀವಿ? ಮೆಗಾಗ್ಯಾಮೆಟೋಜೆನೆಸಿಸ್ ಅಂತ ಕರಿತೀವಿ. ಇದು ಹೆಂಗಾಗುತ್ತೆ ಅಂತ. ಸೊ ಹೆಂಗಾಗುತ್ತೆ ಅಂದರೆ ಆಸ್ ವಿ ನೋ there is formation of 4 megaspores. Out of 4 megaspore, the 3 megaspore they degenerate, the remaining 1 megaspore they become active and function. So this active megaspore enlarges, ದೊಡ್ಡ ಆಗುತ್ತೆ. ನಂತರ the nucleus of the megaspore undergoes mitotic division. They undergo 3 mitotic division. ಯಾವ ಥರ ಡಿವಿಷನ್ ಆಗುತ್ತೆ ಅಂದರೆ here only the division of the nucleus is seen. As a result, there is formation of 8 nucleus. Formation of 8 nucleus. This 8 nucleated cell is referred as the embryo sac. So here, the 8 nucleus. So ಏನಾಗುತ್ತೆ ಅಂದರೆ 8 nucleus ಏನಾಗುತ್ತೆ ಅಂದರೆ they going to organize ಆಗುತ್ತೆ in a definite manner. So ಯಾವ ಥರ organize ಆಗುತ್ತೆ? The 3 haploid nuclei move towards the chalazal end. ಎಲ್ಲೋಗುತ್ತೆ ಅದು chalazal end ಕಡೆ ಮೂವ್ ಆಗುತ್ತೆ. ಮೂವ್ ಆದಮೇಲೆ ಏನಾಗುತ್ತೆ ಅಂದರೆ they get surrounded by cytoplasm and cell wall is laid down around the cell, resulting in formation of 3 antipodal cells. So in the chalazal end there is formation of 3 antipodal cells. The next 3 haploid nuclei ಅದೇನಾಗುತ್ತೆ ಅಂದರೆ they move towards the micropyle end and they form egg apparatus. This egg apparatus is made up of 2 synergids and 1 egg cell. The remaining is only 2 nuclei. The 2 nuclei they remains in the center of the cell and they form a central cell having a 2 nucleus. Thus, a typical angiosperm embryo sac will have 7 cells and 8 nucleus. They will be having 7 cells and 8 nucleus. Okay, now we'll study the next concept called the pollination. Pollination ಅಂದರೆ ಅದು the process of transfer of the pollen grains. ಎಲ್ಲಿಂದ ಎಲ್ಲಿಗೆ ಹೋಗುತ್ತೆ ಅಂದರೆ ಫ್ರಮ್ ಹ್ಯಾಂಥರ್ ಟು ದ ಸ್ಟಿಗ್ಮಾ. ಹ್ಯಾಂಥರ್ನ ಸ್ಟಿಗ್ಮಾಗೆ ಹೋಗುತ್ತೆ ಇದು. which can be from the same flower or different flowers born on the same plant or of different plant. So pollination andre pollination is a process of transfer of the pollen grain from anther to stigma of same flower or different flowers born on the same plant or on different plant is called pollination. Next we we will study the classification of the pollination. Classification ಬಂದಾಗ we usually have two categories of pollination here, as you know right from your schools: the self pollination and cross pollination. Self pollination ಅಂದ್ರೆ ಏನು, cross pollination ಅಂದ್ರೆ ಏನು? Self pollination ಅಂದ್ರೆ it is the pollination that takes place in the same flower of the same plant. Cross pollination andre it is a pollination that takes place from different plants of same species that leads to genetic recombination. Self pollination bandaga again we classify into two categories here: autogamy anta, geitonogamy anta. Autogamy andre ಏನು? Autogamy andre it is a pollination that takes place within the same flower is called autogamy ಅಂತ ಹೇಳ್ತೀವಿ. Geitonogamy andre ಏನು? Geitonogamy andre the pollination that takes place in different flowers on the same plant is called geitonogamy. Under autogamy we have two conditions here. The first condition is called ಕ್ಲೆಸಿಥೋಗ್ಯಾಮಿ ಅಂತ ಹೇಳ್ತೀವಿ. ಎರಡನೇ ಕಂಡೀಷನ್ ಕ್ಯಾಸ್ಮೋಗ್ಯಾಮಿ ಅಂತ ಹೇಳ್ತೀವಿ. ಕ್ಲೆಸಿಥೋಗ್ಯಾಮಿ ಅಂದರೆ ಏನು, ಕ್ಯಾಸ್ಮೋಗ್ಯಾಮಿ ಅಂದರೆ ಏನು? ಕ್ಲೆಸಿಥೋಗ್ಯಾಮಿ ಅಂದರೆ ಇಫ್ ದ ಪಾಲಿನೇಷನ್ ಟೇಕ್ಸ್ ಪ್ಲೇಸ್ ಇನ್ ದಿ ಕ್ಲೋಸ್ಡ್ ಫ್ಲವರ್ ಇಸ್ ಕಾಲ್ಡ್ ಕ್ಲೆಸಿಥೋಗ್ಯಾಮಿ ಅಂತ ಹೇಳ್ತೀವಿ. ಆ್ಯಂಡ್ ದ ಫ್ಲವರ್ ಇಸ್ ಕಾಲ್ಡ್ ಕ್ಲೆಸಿಥೋಗ್ಯಾಮಸ್ ಫ್ಲವರ್. ಕ್ಯಾಸ್ಮೋಗ್ಯಾಮಿ ಅಂದರೆ ಏನು? ಕ್ಯಾಸ್ಮೋಗ್ಯಾಮಿ ಅಂದರೆ ಇಟ್ಸ್ ದ ಪಾಲಿನೇಷನ್ ದಟ್ ಟೇಕ್ಸ್ ಪ್ಲೇಸ್ ಇನ್ ದಿ ಓಪನ್ ಫ್ಲವರ್ ಇಸ್ ಕಾಲ್ಡ್ ಕ್ಯಾಸ್ಮೋಗ್ಯಾಮಿ. And the flower is called casmogamous flower. Now, if you look in the cross pollination, in the cross pollination we have a type called hologamy anta. So hologamy andre it is a pollination that takes place in different flowers of the same plant or different plant is called hologamy. Okay, so today I have just given an outline classification. In my upcoming session I will discuss these pollination in detail. Okay, so thank you, and part 3 will continue in my next session.